is life? Seems like a rather absurd question. I mean, if I had to ask you whether this adorable dog or this crazy looking squirrel was living, I'm pretty sure you'd all know it was the dog. But still, trying to define life in one easy sentence has proven difficult, and so instead, life is characterized by the things it does rather than the things it is. During this session, we're going to try and understand what defines life. We're also going to talk about the different levels of biological organization. We're going to talk about scientific theory, how does science work, and look at experimentation. Here we have 6 characteristics that all living organisms will possess. Firstly, all living organisms are composed of one or more cells. Secondly, living organisms are able to reproduce, and they do this using DNA. Thirdly, living organisms require an input of energy, and they're going to get this energy from their environment. Fourthly, living organisms are able to sense their environment around them. Fifthly, living organisms are capable of homeostasis. And lastly, living organisms are capable of evolution. Let's have a look at each of these characteristics in a little bit more detail. Firstly, the cell. All organisms will be composed of at least one cell. If they're composed of just one single cell, we refer to them as unicellular. If they're composed of more than one cell, they're known as multicellular. Cells are the basic building blocks of all life. Non-living organisms will not be composed of cells. Reproduction is the ability to produce offspring, and this is a key characteristic of living organisms. DNA plays a vital role in reproduction. It houses all the genetic information needed to form the organism, allow the organism to function, and ultimately reproduce. DNA is a molecule that really unifies all life. All organisms are using DNA, and in fact, they're reading that DNA and producing proteins in a similar way. That being said, we could take a human protein, we could stick it in a bacterial cell, and that bacteria could actually produce the human protein for us because they know how to understand our DNA. This fact supports evolution, indicating that we all evolved from a common ancestor with modification over time. We have 2 basic types of reproduction, sexual and asexual reproduction. Sexual reproduction requires 2 individuals coming together, fusing their genetic information with one another. Asexual reproduction just requires one individual making a copy of themselves. The genetic information does not change from one generation to the next during asexual reproduction. All organisms require an input of energy. They need this energy to grow, to function, to reproduce, replace any damaged cells. And there's 2 different ways that organisms can obtain this energy. Our autotrophs or producers, they're going to obtain their energy from the non-living parts of the environment. For example, plants using sunlight during photosynthesis. Our heterotrophs or consumers obtain their energy from the living or once living components of the environment, thus feeding on other organisms in order to obtain their energy. Living organisms are able to sense their environment and respond to the things that they are sensing. So for example, as humans, we can smell, we can see, we can taste, and all those senses help determine our actions. You might think to yourself, well, what about plants? They can't see, they can't smell, but in fact they can still sense their environment. For example, if you had to place a seed into the ground, it's very important that the shoots come up above ground where there's going to be sunlight so they can photosynthesize, and the roots go below ground where there's going to be access to water and nutrients. How does the seed know which way is up? Well, it can sense gravity, and by sensing gravity, it knows which direction to send the shoots and which direction to send the roots. Living organisms are capable of homeostasis. Homeostasis is basically maintaining a constant internal environment despite changes in the external environment. For example, maintaining a constant body temperature or constant salt and pH concentrations within our cells. Think about it as humans. Our body temperature is 37 degrees Celsius. If the environment around us starts to get much hotter than that, our body temperature will begin to rise. And then processes will be put into place to decrease that body temperature. So we'll begin to sweat, and as we sweat, that sweat will evaporate, cooling our bodies back down to 37 degrees Celsius. On the other hand, if it gets really cold, our body temperatures will begin to decrease, and so we'll start to shiver in order to increase that temperature back up to 37 degrees Celsius. Groups of organisms are able to evolve. This is a unifying fact in biology. It's very important to understand with evolution that it's not individuals that are evolving, but it's actual groups or populations of individuals that change over time as the composition of genetics within that population changes over time. Let's have a look at the different levels of biological organization. The smallest level we find is the atom, and in fact, non-living components of the environment are also going to be composed of atoms. Atoms are things like carbon, hydrogen, nitrogen, and oxygen, and these atoms are going to bond with one another to form molecules. So in this case, we formed a molecule of DNA. Molecules are then going to bind and interact with one another, and we're going to start forming different cells within the individual. So in this example here, we have a neuron cell. When cells have a similar function, they'll start working together to perform that function. And this is the level of the tissue. So over here we have the nervous tissue that's going to be composed of a whole bunch of different neurons all working together. The next level of organization will be the organ. So we'll get different tissues that are going to collectively work together to perform a broader function than each original tissue would perform. So in this case, we have the organ being the brain. Our next level of organization is the organ system. So we'll get different organs that are now coordinating with one another. In this case, we're talking about the nervous system. Other systems you may have heard of: digestive system, endocrine system, respiratory system. These will all be made up of specific organs all functioning together to perform an even broader function. The next level of organization is our individual. So we get all these different organ systems that are going to be working together to form that individual as a whole. When we get individuals that are of the same species and they're living together in the same environment, they're interacting with one another, they're breeding with each other, this is what we call the population. The next level of organization after population is our community, and the community is going to have different species living in the same environment interacting with one another. After our community, we form the ecosystem. So in this case, the ecosystem is our tropical coral reef, and the ecosystem is going to include all the living components of their environment as well as the non-living components of the environment. So things like the fish, the coral, the snails, as well as the non-living components such as the water, the sunlight, and the salt. Our next level of organization is the biome. Now, biomes are regions of our planet that have similar climate, and because they have similar climate, they have similar organisms inhabiting that area. So for example, think of an ocean. All right, we see oceans worldwide. They have similar organisms living in them. Other biomes would be things like deserts, tundras, and rainforests. Once all these biomes come together, that's when we form the biosphere. So the biosphere is our whole Earth, including the atmosphere where life is possible. Okay, let's tackle the next big question. What is science? When you think about science, what comes to mind? Once again, science is a very broad topic that's difficult to narrow down and define. Personally, I like the sentence as it incorporates many of the aspects of science. So the sentence states that it's an objective, self-correcting method for gathering and organizing information about the natural world through repeated observation and experimentation. So that's quite a mouthful. And let's break it down. Firstly, and very importantly, science deals with the natural world. It cannot deal with the supernatural. So science cannot answer questions about religion or zombies or fairies. It can only answer questions about the physical natural world around us. Secondly, science involves observation. So when we observe things in the world around us as humans, we start to question, why does this happen? What happens if we change things? And it's that observation that sparks science. Not only does it spark our interest, but it's going to be needed to answer our questions. Science involves experimentation. So once we've observed something, we go into the lab and we perform experiments. Experiments are important because they allow us to restrict our parameters and look at one parameter at a time. That way, when we get our results, we know that the results are due to that specific parameter and not other confounding things. Very importantly, we can't just observe something once or do one experiment and think that we know the answer. It has to be repeated over and over again, not just by one individual, but by different scientists around the world before we can actually start to figure out what's going on. The next important thing about science is that it's objective. It's not based on your own personal beliefs or bias, but has to be completely objective. Science is also self-correcting. So as we perform more experiments, as we do more observations, we can start changing our ideas. For example, we originally thought the world was flat. But as we've gained more information and more knowledge, we've realized that our globe is spherical. In science, it's also really important that we gather the information that we obtain through our experiments and observations, and we organize that information so that other people may have access to it and may build on top of it. So very importantly, science needs to be open and accessible to everyone. And it needs to be open to challenge by anyone at any time as long as there's sufficient evidence. The scientific method, or the process of science, is how we generate scientific knowledge. It's not just one recipe, but it's more of a core logic of how science works. The process of science starts off with an observation. So we see something in the natural world around us. And this will naturally lead us to question, why is this happening? What makes it happen? What can we do to change it? From our observations, we can come up with a hypothesis. That hypothesis is just a bold statement about what we think is going on. It's really important that that hypothesis is something that we can test, either through observations or experimentations. It also needs to be completely objective and not based on our own personal biases, and it needs to be falsifiable. So it needs to have a possibility that it could be proven incorrect. Once we've come up with a hypothesis, we can then form a prediction. A prediction is an if-then statement. So if we do this, then something else will occur. Once we have our predictions, we can then test those predictions, either through experimentation or observations. If those experimentations or observations support our hypothesis, then we'll go back and find more experiments, more observations, in order to gain further and further support for that hypothesis. If, however, our tests reject the hypothesis, we'll go back to the hypothesis. We can either revise that hypothesis in some way, or we can come up with a new hypothesis. So let's say, for example, you live in an area where you have both a lake and the ocean. So you look outside your kitchen window and you have this beautiful lake, and you look outside your bedroom window and you can see the ocean. So you wake up one morning, and when you look outside, you see that the lake is frozen over, but the ocean is not. So that is your observation. And that will lead you to question, well, why did the lake freeze over? Why did the ocean not freeze over? And now you can come up with some kind of hypothesis. Let's say that you think that it might have something to do with salt concentration. So you know that the ocean is super salty, whereas the lake is fresh water. Remember that your hypothesis needs to be a bold statement. So you can say something like salt water freezes at a lower temperature than fresh water. The next thing you would do is come up with a prediction. Let's say in this case your prediction was, if I place salt water and fresh water at 0 degrees Celsius, then my fresh water will freeze, but my salt water will not. And now it's time to perform your experiments. You could obtain 2 beakers and place water in both beakers. Add salt to the 1 beaker. And place both beakers at 0 degrees Celsius overnight. You would then come back the next morning and observe the beakers. Let's say that when you came back the next morning, the fresh water was frozen, but the salt water was not frozen. Now this provides support for your hypothesis. You could not say that you have proven your hypothesis, but you could say that you've gained support for the hypothesis. You would then go back, find more experiments that you can run to gain further support. Perhaps you could change the temperatures that you're conducting the experiment at. Perhaps you could change the salt concentration you're conducting the experiment at, just gaining further and further support for your hypothesis. If, however, you came back the next morning and either both beakers of water or neither beakers of water were frozen, now you would have rejected your hypothesis. So you would have to go back, revise your hypothesis in some way, or start with a new hypothesis. Maybe you want to look at something different, like the size of the body of water instead of the salt concentration. Let's have a look at this example. Let's say that you go to use your flashlight and it's not working. This observation would lead you to question, why is my flashlight not actually working? And then you could come up with a hypothesis. Perhaps you'd hypothesize that maybe the bulb is broken, or maybe the batteries are flat. Remember that you can only test one hypothesis at a time. So let's deal with the first hypothesis, that the batteries are flat. Your prediction would be that if you replace the batteries, then the flashlight would work. So you would go ahead, conduct your experiment, you would change out the batteries, and when you go to try and put your flashlight on, it doesn't work. So now your experiment has refuted your hypothesis. Now you could go back, revise your hypothesis, and look at the second one, where it might be the bulb. Your prediction now would be that if you change the bulb, then the flashlight would work. So you run your experiment, you change the bulb in the flashlight, and your flashlight begins to work. Remember that you've just gained support for your hypothesis. You cannot say that you've proven your hypothesis. In this case, perhaps there was something else involved that you didn't even detect. Perhaps when you were fiddling around with the batteries and the bulb, you connected a loose wire without even knowing it. So it's important to remember that we never say that we prove a hypothesis, rather we either support or reject that hypothesis. Let's talk a little bit more about experiments. An experiment is an investigation that you conduct in a controlled environment, where you control factors so that you can look at one parameter at a time. The important thing about experiments is that it must be able to be repeated by the original person who conducted the experiment, as well as by other scientists around the world. When designing an experiment, one parameter that needs to be thought of carefully is sample size. The larger the sample size, the better, because you're going to get a better idea of what's going on. However, sample size is often restricted by funding and the availability of resources. You need to be able to find that balance between getting accurate results and staying within your funding and resource limitations. When designing experiments, there are some important parameters that need to be considered. We have different variables. The independent variable is the one that you are going to manipulate. The dependent variable is the one that's going to change depending on what you do to that independent variable. Standardized variables is everything else that remains exactly the same, so that you are just looking at one specific variable at a time. The other important factor to consider when conducting experiments is controls. Controls provide a baseline of comparison so that when you get your results back, you can compare them to something else and know whether there has been an impact or change. Pretend that you took a trip to the Amazon rainforest. When you got there, you encountered a tribe that had a very low incidence of cancer. You also observed that this tribe feasts on a wildflower daily. This may lead you to question, is there something in this wildflower that can prevent or cure cancer? Your observations and questions may lead you to a hypothesis, and you may want to look at one compound within that flower at a time. Let's call it compound A. Now you can come up with your hypothesis. Say, for example, you hypothesize that compound A will kill cancer cells. The next step would be to form a prediction. If cancer cells are given compound A, then they will die. Now it's time to conduct your experiments. You go into the lab, you grow your cancer cells, and then you administer compound A to your cells. When you come back the next day, you notice that your cells have died. However, there's a flaw in your experiment. You didn't set up any controls, so there is no way to be certain whether the cancer cells died because of compound A or because of another factor that was occurring during your experimentation. You therefore repeat your experiment, this time including controls. You decide to set up two controls. One control is just the cancer cells. You do not administer any kind of compounds to it. This would be known as your negative control. The next control you set up is known as the positive control. To this control, you give a compound that you know for certain kills cancer cells. When you come back the next day, you're able to now compare the results from compound A to your negative and positive control. This way, you're able to know whether cell death was a result of compound A or due to something else occurring in your experiment. You're also able to compare the toxicity of compound A to a known compound such as compound B. Looking at the different variables in this experiment, your independent variable would be the compound administered, whether we have compound A, whether we have nothing, or whether we have compound B. Your dependent variable would be cancer viability, so do the cancer cells actually survive? Your standardized variables would be everything else. This would mean you'd have to keep the same incubation time, incubation temperature. You would have to start off with the same number of cancer cells. You would have to grow them in the same kind of dish. Make sure you feed them the same kind of nutrients so that everything else is exactly the same. This way, when you get your results back, you know that they're due to compound A and not due to something else, such as temperature or time. We've come to the end of our session. Please go ahead, pause your video, answer these questions, and then unpause so we can go through the answers together. Firstly, what are 6 characteristics of living organisms? Well, they're composed of 1 or more cells. They reproduce using DNA. They're capable of homeostasis. They're capable of evolving. They are able to sense their environment and respond to it, and they need to obtain their energy from their environment. What are the levels of biological organization, starting at the smallest? So we have atoms, then molecules, then cells. We then form tissues, organs, organ systems, after which the individual is formed, then populations, so individuals of the same species. We then have the community, the ecosystem, the biome, and then the biosphere. Why are experiments important? Well, experiments are important because we're able to go into a very controlled setting where we can look at just one parameter at a time, ruling out any confounding factors. What are the steps of our scientific method? So we start off, the very first step is observation. Observing something leads you to question. You then form a hypothesis. After you've developed your hypothesis, you come up with a prediction, which is your if-then statement. You then run your tests, which can either support your hypothesis, after which point you'll find more tests and experiments to conduct to gain further support, or if your tests refute your hypothesis, you can go back, revise the hypothesis, and go through the process once again. When talking about experiments, what is the dependent variable? The dependent variable, I like to think of it as it depends on the independent variable. So your independent variable is whatever you're going to be manipulating, and as you manipulate it and change it, so you will see changes in your dependent variable. Your standardized variables, that is every single thing else that's going to be kept exactly the same and constant. And then lastly, your control. Your control is very important. It's going to set up that baseline so when you get your results, you can compare your results to your control and see if there's been any kind of effect. Thanks for watching, and I'll see you guys next time.